So my name is Jess Levicki. I'm an assistant professor at Brown University in Providence, Rhode Island. My lab focuses on how environmental exposures and genetic mutations impact development, and we're specifically interested in the development of the heart and brain and the intersection between brain and heart health. So as a developmental biologist, I think that, we're trained about the importance of different genes and their roles in forming organs and how, small mutations can impact health. So we think about things very much so from that genetic perspective. Scientists will often use small molecule screens for, drug discovery, viewing it from a therapeutic potential, also looking at it from a loss or gain of function perspective. People don't think about environmental health as, a driver of human disease, the same way that they think of, genetics. Something that I took from my undergraduate training was this idea that if you look at questions from multiple levels of analysis, you'll have a richer understanding of those processes. And so when we are looking at something, we want to look at, a molecular level, a cellular level, a functional level, and one of the best functional levels is looking at behavior. We use DanioVision, so we have two different behavioral units, and we use those behavioral units, I think, in some classic ways. We tend to use the light-dark assay to assess the impact of different environmental chemicals on behavior. So we're looking at, environmental contaminant exposure in Campari fish, and then using our Noldus units to photoconvert the protein, and then we do our image analysis using the confocal. Well, zebrafish are wonderful. I think anyone who's worked with zebrafish feels pretty passionately about them. They're a tiny aquatic model system. They're initially transparent. You can extend that window of transparency with pigment inhibition, but also through, you can have genetic mutants that make them easy to image. So we're a very imaging focused lab. And so it's really satisfying to see cells migrating or organs forming or looking at, the function of cells in real time. And so there's just a huge amount of beauty in that, as well as, scientific power. So, one of the things I think is really nice about the Noldus products that we use is the fact that we can look at, larval behavior at different time points with different types of stimuli. We can look at adult behavior. So that's been really a wonderful way to integrate our behavior, the units, as well as optogenetics into our research questions. There's so many different things that go into setting up a lab. I would say what really stands out to me is the development of a strong team. So having a group of individuals that are fundamentally excited about science and are, willing to work together for a common cause, even if they're not necessarily working on each project together, have individual projects, but to be able to be collaborative, since collaboration is such an essential driver in science.